There are different types of point mutation. If you take the transition mutation, the sixth position of wild-type gene, AT base pair, is replaced by a GC base pair. That is, one purine pyrimidine base pair will be replaced by another purine pyrimidine base pair. And in transversion mutation, the purine pyrimidine base pair is replaced by another pyrimidine purine base pair. That is shown in the red color. That is, AT is replaced by TA. And in insertion mutation, a base pair inserted between the other two base pairs in the wild type gene. Here, a GC base pair is inserted in between AT and CG base pair. And in deletion mutation, one base pair is deleted in the wild type gene. And here, an AT base pair is deleted. You have to see that not all point mutations are necessarily lethal. Transitions and transversions are relatively harmless because they result in replacement of only one amino acid in the peptide chain coded. Even after the defective protein is still functional, such a mutation we call it as silent mutation. Insertion or deletion mutation causes the entire DNA beyond the point of the mutation to misread. Such mutations are often lethal because it causes a change in the reading frame of the DNA and causes the death of the organism processing it. Next is about back mutation and suppressor mutation. The mutation of wild-type gene to a form that results in a mutant phenotype is usually referred to as forward mutation. Wild-type and mutant represent two different phenotypes. For example, if we consider the two allele controlling brown and blue eye color in humans, occasionally a few adjacent nucleotide pairs within the gene control the sequence of color in humans. However, in a population composed almost entirely of brown-eyed individuals, the allele for blue eyes might be thought of as a mutant allele. That is, a subsequent mutation may occur that restore the original wild-type phenotype after the next generation. This is referred to as back mutation or reverse mutation or reversion mutation. Reversion may occur in two different ways. Restoration of the original phenotype may occur either by a true back mutation at the same site in the gene of the original mutation, restoring the wild-type nucleotide sequence, or by the recurrence of a second mutation at different location in the genome, which somehow compensates for the first mutation. Mutations of the latter kind are called suppressor mutations, that is, they suppress the effect of the original mutation. Suppressor mutations may occur at distinct sites in the same gene as the original mutation or in different genes, even in different chromosomes. Next is effective point mutation on protein synthesis. And the changes occurring may be due to same-sense mutations, missense mutations, nonsense mutations, and frame mutations. In this, about the same-sense mutation, although mutation has changed the nucleic acid sequence of a codon, the altered codon still encodes the same amino acid. This is possible because the code is degenerate, thus the same-sense mutations are silent. There will be no change in the synthesized protein. In the second type, missense mutation, a single base pair substitution changes a codon for one amino acid into a codon for a different amino acid, resulting in an amino acid substitution in the polypeptide. And third one, nonsense mutations, a base pair substitution changes the codon for an amino acid into UAG, UAA, or UGA. They are the stop codons. Therefore, the premature termination of the translation occurs so that only inactive polypeptide fragment is produced. And the fourth one is the frame shift mutation. These are due to the addition or deletion of one or two base pairs. This shifts the reading frame so that all codons from the one containing the mutation end of the message are read out of phase and encode incorrect amino acids. This in turn results in the premature termination of translation or the protein synthesis. What are spontaneous mutations? Mutations that occur without a known cause are called spontaneous mutations. They may be truly spontaneous, resulting from an inherent low level of metabolic errors, that is, mistakes taking